Hello, everyone. This is Kenan, and I'm a PhD student at Izmir Democracy University, Electrical Electronics Engineering Department. In here, I would like to present the work that we've been doing at Izmir Democracy University Lab, the Computer Vision Lab, and what further work we aspire or we will continue to be doing. The work I'm going to be presenting, or the work that we've been doing, is related to finding an automated classifier for COVID-19 diagnosis using computer tomography images. All right, let's go ahead. So let me give a brief introduction of who I am. I've got a bachelor and a master's degree in control and automation. I focused mainly within those on probabilities, signal processing, artificial intelligence of both structured and then unstructured data. I used R, I used MATLAB, and recently I use Python. I also work as a vocational teacher, Engineering Science Unit 3 for PTech or Person, right? So undergraduate level teaching, vocational teaching. I started my PhD 2 years ago. I focused on machine learning and focused on image processing, computer vision, and mainly I got involved practically with COVID-19 diagnosis through workshops, one that I'm going to be presenting here, a project I'm going to be presenting here. I also speak languages. I learned 2 languages professionally, and I speak 2 other languages to a limited level, along with my native language, of course. What I aspire to do in the future is to improve my engineering skills mainly. I'd like to conduct in-depth research in the area and also improve my programming skills as well. I hope to stay in the medical domain or other relevant domains like microscopies and other relevant domains of images. Perfect. Now the content of the presentation is going to go like this. First, we'll start with a very relevant literature review, recent and relevant in COVID-19, and then I'm going to introduce the data set, COV19D database. I'm going to introduce the current methodology, the achievements that we've got in terms of COVID-19 diagnosis with that methodology, and then I will end with a conclusion and further research work that is to be done still, acknowledgement and references for sure. All right. So there is a lot of work that has been done in the area of the COVID-19. Mainly, I'm talking about images. So there are different image modalities, CT, X-ray, and other modalities. But I'm going to be discussing CT and X-rays in here in my literature review. Mainly, I'm going to focus on just narrow recent ones. There is the paper by Sarah Atito. I hope I'm pronouncing the name correctly. This introduced a deep neural network called IST-COV-NET. This provides an evaluation of 2 different 2D and 3D approaches on 2 different data sets, actually. I'll present those data sets in the next slide. The final model here combined a attenuation mechanism with a slice level combination. They used long short-term memory in there, an extended architecture for 3D data. You can refer to the data set. The results were good. It was so good, actually, that it's being used now in a hospital in Istanbul. They have a collective data set that they called IST-C data set, right? So this is one research. And then there is the baseline. The baseline approach in here is something that happened recently as well with regard to COV19D database, the database I'm going to be working on. So this will give us the baseline for the work, right? So basically, we need to get results that are better than the baseline results, mainly in terms of the macro F1 scoring here. So the baseline here is represented by Dr. Dimitris and others. And they used a CNN recurrent neural network architecture as a deep neural network model for the results. We'll discuss the results and compare to them in the coming slides, of course. My research work in here further explores the same data set, COV19D database, and introduces a less hand-engineered and efficient deep learning-based solution for COVID-19 diagnosis from this data set. The results will exceed the baseline's performance and other alternatives provided through an international competition here. All right, let's go ahead. So I'm going to present some data sets, right? The data sets used. I'll start with an X-ray data set, chest X array 8 data set. This has like a frontal view X-ray images of 32,717 unique patients, right? So this is the number of the patients, quite big number. Each image can have multi labels, right? So the image can have different labels. In general, the number of X-ray images will be 108,948, right? With multiple labels, as we said. In terms of the CT images, there is the IST-C data set, right? This is a CT scan data set. It has COVID-19, normal, and other, right? So these are the labels. CT scans from 1 hospital. The number of the CT scans is 712. The number of the COVID CT scans is 336 in this data set, and the rest, the normal and the others, are 376. So we have 336 COVID cases in this data set. The other data set, which was also used by Sarah, the same paper, the data set called MOS-MED data, right? So this is also a CT scan data set, indicated COVID severity in here. So you have levels of severity, 4 levels, right, of the CT's data. So the number of the scans is 1,110, and the number of COVID is 856. 254 are not COVID related CT scans. Now the data set that used in this study is COV19 CT database, which is labeled either COVID or non-COVID, right? So it's a binary classification, or binary labeling. So COVID-19 and non-COVID-19 CT scans, it's for 5,000 patients, right? 5,000 CT scans. You have a training validation. You have also test partition. Train and test set, we have 855 for COVID cases and 1,079 for non-COVID cases in COV19 CT database, right? So quite big, as you might see. Okay, so this is a little comparison of recent COVID-19 related data sets. All right. So let me discuss here why we in our lab chose the COV19 CT database, the last one. It's a novel database, right? It started last year in the mid of 2020 through an international competition. We got to know the people in there. We got to know those practitioners who labeled the data set a little bit. Then we chose it because it's a new data set. Second is that it's a challenging CT images in this data set. Looking at the slides, you can notice the variety of numbers. So in one CT scan, you might see you might find like very few number of slices, like 1 sometimes, or the number could be very, very high. Like, so this variation makes it more challenging in this. So this challenging nature is another reason that we chose the data set. A third reason would be that it's a big data set, which makes the work more robust, right? So the results that we find are more robust to variations, if you want to say, and since you are building your model on a big data set, then you are kind of like guaranteeing good results in a way, or we hope. All right, so these are 3 main reasons why we chose the COV19 CT database for our research. All right, so what is this database? Basically, it was published for the purposes of developing an automated classification algorithm for COVID-19 and non-COVID-19 diagnosis. And it consists of about 5,000 patients, as I mentioned in the table before. It corresponds to a high number of patients, more than 1,000 patients in here. The annotation of each CT slice has been performed by 4 experienced, each with over 20 years of experience, right? So very experienced medical experts. We have 2 radiologists and 2 pulmonologists. Labels were provided by the 4 experts, showed a high degree of agreement. So there's a great degree of agreement between the experts, around 98% agreement. And it was never created as a result of positive PCR test or something like that, right? So it's very much by experts, these labels. All right, cool. So this is the data set. Now I'll get you introduced to partitions of the data set. So you have the training partition, or the training set, which consists of 690 COVID cases, 870 non-COVID cases. There's also the validation set, which has like 165 COVID cases and 209 non-COVID cases. Finally, you have the unseen images, or the test partition, if you want to call it. Of course, here the labels are not provided to the teams, or are not provided very much to us. You have to send your results in there to check with the labels of them. So you have in a test partition 3,455 images or CT scans. All right, the CT scans include different numbers of CT slices, ranging from 50 to 700. The labels for the test partition, of course, are not provided. You have to send your results there. What is required basically from the work in here is to get high macro of 1 score. So you want to try your method or your methodology on a validation set first, get a high score, high macro of 1 score, and then you want to send your results to check it on a test set, the unseen images for COVID and non-COVID diagnosis, right? All right, so this is basically what the data set is about, the sets of the data. All right, now what have I done so far in terms of that project? I presented a less complex deep neural network to achieve COVID-19 diagnosis from the CT images, right? We'll check on that. I showed that processing the CT images with a region of interest, so focusing on the region of interest and localizing that, localizing the work of the neural network on the region of interest dedicated to the long areas will improve the diagnosis performance of the model. I proposed taking patient level diagnosis from slice level processing via different voting methods, right? So what I worked on the 2D slices, right? And then there is a proposal of how to take a patient level on a 3D level. I use different voting methods for that. I'll present that as well. I evaluated the performance of the proposed solution on a recently relatively large challenging data set, right? So there's the test partition, there's the validation partition, the COVID-19 CT data set, and I presented my results, or I evaluated my results on those, on this large data set. Perfect. So the methodology being followed in our lab, I'm going to work with, start with slice processing techniques. So the techniques that we use to process the slices themselves. So as you can see here on the right-hand side of this slice, there is a region of interest, right? So a fixed size rectangular shape will localize the region of interest with those two lungs, the right lung and the left lung, right? So the lung regions in a slice were more focused in the work, right? So there's this cropping of this area of each of the slices in the CT scans. It contains, as you can see again, the left lung and the right lung. All right, so this is one step for slice processing. And then methodology being followed also is that those cropped images were then blurred using Gaussian filters, right? So you can suppress the noise and enhance the structure of the images using the Gaussian filter. And then I used histogram binarization on the results, right? So you can see here the binarized image through the histogram. So this histogram will give us a binarized images through a threshold, right? So I'm looking at the histogram of these images, right? I'm taking the images and then I'm looking at the histogram, and then with a threshold you can get the binarized images that you see on top. The histogram based threshold was applied mainly to remove the uppermost and lowermost slices of the CT images, which we consider to be non-representative of the existence of COVID-19, or non-representative. They will not be showing the COVID-19 case, or they're not going to be. We're not going to be able to detect COVID-19 from those non-representative slices, which will be the uppermost and the lowermost slices of the CT images. So the method to remove those slices was to binarize the slice, right? Take a binarization through the histogram. So looking at the histogram, I chose a threshold. As you can see, the threshold that fit after normalizing the pixel values was 0.45. So based on this histogram, I will be removing the slices, the uppermost and lowermost slices in the CT images. How did I do that? Now, the binarized images pixels were used to find the CT volume level threshold to remove those slices. So what basically happened is that I chose randomly 4 slices in a training set, right? CT scans 5, 6, 7, and 8. those slices were chosen. And then in this figure in here, you can see the number of white pixels in the slices left after binarization. And then three levels or 3 degrees of importance were given to those to the slices, right? So it depends very much on the number of white pixels in each slice. So if the number of white pixels in this slice, in the binarized sliced image, if you want to say, were less than 4,500, then this slice will be considered non-representative. Like, as you can see, it's a level 1, which means it doesn't represent, right? So we have 1, 2, and 3 level of importance for the slices. So basically 3 is very important, right? It has this dark area that shows you the COVID cases or the COVID issue or the COVID problem in the CT image. But if it's a level 1 or level 2, probably level 1, you want to remove these slices. So you can tell from there that if you choose some kind of like a flexible threshold, that you can remove those slices that has less than 4,500 white pixels in them. You also want to be careful here not to remove all the slices in one CT scan, right? So this is a very important issue. That's why I used a little less of a threshold, right? I couldn't like make it a little higher, because in that you might end up with some CT scan with no slices at all, and thus there will be no decision or no voting decision made for that patient. All right, perfect. So using this methodology, the number of the slices were reduced by 16% reduction on a training set and 15.3% reduction on the test set, right? So the number of all the slices in the training set were reduced by 16%, and the number of all the slices in the validation set were reduced by 15.3% using this technique. Perfect. So this is slice processing techniques. Now, all these images that the scan of images, the CT scans, after removal of uppermost and lowermost of the CT scan, and after cropping of those images, you want to put it into a deep learning model. So here I use the convolution neural network model, and in here you can see in this image what the structure of the model is. So you have the first start with a convolution layer 512 by 512 and 16. Here we have to be careful is that 512 and 512, this is the original size of the images. Of course, the cropped images did not have this size, and thus it followed a different order, but this is a standard order for the original image. So there's a convolution layer followed by with padding, patch normalization, ReLU activation, max pulling, 2D max pulling as well, to reduce it to 256 by 256 and 16. And then there's another convolution layer, exactly the same thing. And there's the third convolution layer and a fourth convolution layer as well, until you reach here a flatten layer, right, with some number, yes. And then there's the dense, batch normalization, ReLU activation, and dropout. Finally, there's the dense layer that will give you the output, and the unit for it is 1. The output in here, the output of the final dense layer, will be the class 1 probability. Class 1 probability is the non-COVID probability. So the probability that the slice is a non-COVID will be the output of the final dense layer, right? So the size of the images, or the size of the input images after cropping with the rectangular region of interest, will be 227 by 300. So this is the size that will be input to the deep learning model. All right, perfect. So this is the architecture of the CNN model that is used in our work. All right, so with that, also there is hyperparameter tuning in the model. As you can see, the learning rate decays. So there's an initial learning rate, and then the initial learning rate, I use stochastic gradient descent, I should mention that, as an optimizer. And then the decay of the learning rate is computed through the equation that you see in there, decay rate to the power of optimizer step and decay step. The initial learning rate was set to 0.1. And there is 0.96 decay rate was used. So the value of the steps divided by the decay steps is an integer division in here, which means like there is a decay in a staircase fashion. The optimizer steps were defined as the train steps, were the number of the trained samples divided by the batch size. Of course, I tried several batch size. The 128 by size is the one I settled on, right? So this is the number of the trained samples divided by 128. The validation steps also is the number of the test or the validation samples divided by 128, right? So this is how the train steps and the validation steps were taken, and the decay steps were set every 100,000 steps. On the other hand, there is also usage of class weights, right? So because it's the number of the COVID and non-COVID in the training set is unbalanced, or you can say that the data is imbalanced data set, and then the ratio is 1.197 to 1, right? COVID to non-COVID. So there's the class weight usage while training, which is the number of the images for a class in the training set divided by the number of all the images in the training set, right? So this is how I assign the class weight. All right, perfect. So this is about hyperparameter tunings and the class weight as well. Moving on. All right, so diagnosis at the patient level and the slice level, actually. So as I said, the final output from the dense layer of the deep learning model is the class 1 probability. The class 1 probability is the probability of that the slice is a non-COVID slice. So if the class 1 probability is greater than class probability threshold, then I will predict the slice as a non-COVID. Otherwise, I will predict the slice as a COVID. Now, this threshold is chosen. I tried different thresholds, right? So I'm trying to check the probability that the slice is non-COVID to different thresholds, right? So if the probability is high, you want to predict it as COVID. Otherwise, you want to predict it non-COVID. So I tried different thresholds in here to check with the results. So is the probability predicting non-COVID, and different thresholds will be presented in a bar chart in the next slice, right? So this is how I worked on processing the slice level predictions, right? So this is at slice level. As for the patient level prediction, right, to predict whether the patient is COVID or not, the patient is diagnosed based on the presence or absence of COVID slices in their chest CT scans, right? So if the patient CT data contains more non-COVID the predicted slices than the COVID predicted slices, then we will say that the patient doesn't have the COVID. Otherwise, the patient will have the COVID. So this is the majority voting method. So after you get all the slices in one CT scan, you can tell that this CT scan or this patient has a COVID or non-COVID based on the majority voting. And if they're equal, it will be predicted as non-COVID. That's the method that we used in here, right? This is what we did in the lab so far. Perfect. So let's check now. As I said, on a slice level prediction, you have to compare the output of the final dense layer, which indicates the probability that the slice is non-COVID, to different thresholds. And based on that, you can decide that this slice is a COVID or a non-COVID. So here, different thresholds were chosen, right, as you can see. And then the results were checked, right? The final results were checked on the validation set. The results in here indicate the macro F1 score, right? This is what we care about. The macro F1 score is the matrix measure that we used for our work in here, right? This is the measuring our work, right? So the results show here that if we use a threshold of 0.40, we'll get the highest macro F1 score on the validation set, right? So this is at the patient level. We get the highest macro F1 score on the validation set when we use the 0.40 threshold. And thus, this is the threshold used to decide at slices level whether the slice is a COVID slice or a non-COVID slice. The baseline method's macro F1 score was 0.70, right? So this is the baseline. As I said, there's the baseline method introduced by Dr. Dimitris and others. On the validation set, the macro F1 score is 0.70. You can compare to 0.882 in our work, right? So we achieved 0.882 macro F1 score on the validation set, and thus we exceeded on the validation set the results of the baseline. All right, moving on now. All right, so here we can see the results on the validation set in terms of the confusion matrix. So as you can see here, the actuals and the predicted ones, there's of course a misclassification here. There is 13 of misclassified COVID cases was predicted as non-COVID, and there's 30 of actual COVID cases were predicted non-COVID, right? So this one is vice versa. This one is actual non-COVID cases that were predicted COVID. Here, of course, as you can see, there is misclassification, and there's high misclassification, especially when it was COVID. 30 of them were COVID, and then they were predicted as non-COVID. For deployability, further work is indeed needed to figure out how to reduce this number, since predicting a COVID case as non-COVID is more risky or more dangerous than the other way around, right? So, but mathematically for now, this is what the confusion matrix shows in our work on the validation set. Perfect. All right, so here in this slice, you can see the correct classified images. So here, examples of correctly classified slices from COVID-19 on the right. This is correctly classified COVID-19, and this is non-COVID cases which are correctly classified. As you can see from those two slices, this is a misclassified COVID and non-COVID, right? So this is the COVID misclassification, this is non-COVID misclassification. So you could tell that that those are more representative slices, right? Because you can see the dark areas, and thus those can indicate that the person has COVID or not, whereas it's difficult to tell from those ones. And accordingly, our model did not was not very successful in predicting the COVID when the slices were non-representative, as the one you see here in the right. So they are non-representative slices, uppermost or lowermost of the slice, uppermost and lowermost of the CT scan, right? So this is where the misclassification actually occurred. All right, here also you can see the attention results, right? So what the model is focusing on, this is a guided GradCam visualization on the left, the correctly predicted COVID slice. So this is a correctly predicted COVID slice case, and it shows here what the model is paying attention to. The model is quite much paying attention to the area where it's supposed to be paying attention to. So in general, you can say that there is some good performance, or that the model is not very off the right path. Perfect. All right, so here we get to the test partition results. So our method, or the methodology presented in here, achieved 0.82 macro F1 score. And then the macro F1 score for non-COVID was 0.96. The macro F1 score for the COVID cases was 0.68, right? So this table shows here comparison between our model on the test partition and other models, right? So here you've got the baseline model, the one that we described before. So on the test partition, it's 0.67, right? This is the baseline results. And then you have a hybrid deep learning framework, CTNet, that achieved 0.78, another customized or custom deep neural network that achieved 0.78, and then there is our methodology, 0.82, and other CC80 and DWCC that achieved 0.88. Right? So this is a little bit of comparison on the test set between our work and other teams' work or other alternatives on the same data set, right? So, and here is where you can get to the leaderboard. This is a link for the leaderboard if you're interested to see all the teams and all their results on the test partition. All right, so we well exceeded the baseline and other alternatives as well. You could see that more clearly from the leaderboard link. Perfect. All right, so conclusion and research work. My research here led to a solution for COVID-19 diagnosis using deep neural network and image processing techniques, right? So there's processing the images, and there's also deep neural network used to produce the results. In this methodology, using a rectangular region, as you said, as we mentioned, we kind of like localized or focused the attention of the model on the right lung and the left lung as well. So there's a rectangular cropping. So it gave better performance. And this actually proves that limiting the region of interest with the long volumes instead of processing the whole CT images is a promising approach, right? So for further work, this is a very promising approach to focus on the lung area. With that in mind, we can say that segmentation of the lung prior to the classification would be great improvement, should give a great improvement for the diagnosis performance in this method. This research, of course, is still in progress. And it's open to collaboration for improvement. I should probably also mention that there is the second round of the competition, COVID-19 competition, that will be held in the framework of AI-enabled medical image analysis, right? So there is a second round of the competition that is going on this year. Good luck to all the teams in there. Perfect. What we're going to do, of course, we will be resubmitting the paper, summarizing the research work, the paper that you can see the preprint of the paper so far. We'll be submitting this paper. We will focus further on the segmentation methods of the lung. Different segmentation methods should be checked on the lungs. High microFON score is expected to be reached by improving the methodology that has been presented in here through segmentation or other ways. Exploring, we could be also doing exploring diagnosis performance of deep learning methods other than the convolution UNet for segmentation, right? So we might be working on diagnosis performance for deep learning methods using non-conventional UNet or other deep learning methods with attention mechanism, semi-supervised approaches, application of deep learning methods for anomaly detection, and so on and so forth, in help in the direction of this research or similar researches. The research does not have to be applied to COVID-19 diagnosis only. It might be applied to other lung diseases with characteristics similar to COVID, such as pneumonia. So if we can find some work in there that can achieve good results, we can also apply to COVID and to non-COVID as well. Pre-processing and post-processing of the images are also possibilities of further work or further research work in the lab. I would like to, yes, acknowledgement to all the people who helped me and participate in the work, especially Associate Professor Dr. Devrim and I, patience, as well as knowledge. Of course, when you start your PhD or you start your studies, you will be annoying your professors or the experts. You'll be annoying them a lot with the level of knowledge that you have, right? So you have to go with them step by step. Sometimes I would be sending them bugs when you're doing programming, so we'd be sending them bugs where like the program is not working. I don't know where you can go to Stack Overflow or some other areas to find the solutions there. So of course I should acknowledge their patience, not just Associate Professor Dr. Devrim, but all other faculty members at Izmir Democracy University. They have amazing patience and they are amazing experts as well. Third, I would like to acknowledge the work of the medical staff and the others who manually annotated this big data set, COVID-19 CT database, and they shared it with us, with all the other teams to work on the methods, to present their methodologies. Acknowledgement also to the facilities that are provided for doing this research, the workstation, of course, in the lab where this work has been conducted. These are some of the references of the other teams, other papers, other methodologies presented with the same data set, with different data sets that can help in this area as well, to understand this area a little better, along with the link for the competition. This is where the competition website is, right? Thank you very much. That's all for here. You can probably also check the GitHub, my GitHub, and the lab under the organizations in there to check further the work that we've done, or to see how the methodology went on through the codes and stuff. Thank you very much.